All right, welcome to our second Science on Tap for fall of 2020. Folks are still filtering in, coming in from the waiting room. So we are going to give folks a couple more minutes before we get started, but we are very excited to have Dr. Doug Durant with us from the Biological Sciences Department. And I'll do the formal introduction in just a couple of minutes. So welcome, and we're glad to have you tonight.
How you would speak to him and what he was going. They love watching us. I would hope me.
Please leave your microphones muted.
All right, welcome to our second Science on Tap. This is our second go at a virtual remote presentation. We are very excited and lucky to have Dr. Doug Durant with us from the Biological Sciences Department. So just a couple of notes about how our Science on Tap is going to work. We do ask that you keep your microphone on mute so that everyone can hear Dr. Durant as he is presenting. If you have questions along the way, we're going to have plenty of time at the end of the presentation for you to ask your questions, or feel free to go ahead and throw them in the chat. I can moderate these questions as we go throughout the talk. So welcome again to our Science on Tap. Thank you so much to the College of Science for helping facilitate and advertise and get the word out about this wonderful event. Our topic tonight is life and science in the time of coronavirus. Dr. Doug Durant is going to be presenting this evening. Dr. Durant joined the Cal Poly Pomona Department of Biological Sciences in 2015 as an assistant professor, where he's currently investigating how the dynamic regulation of immune cell entry into the central nervous system contributes to both protective and pathological outcomes during viral encephalitis. So his experience as an immunologist really sets him up well to present this evening about coronavirus. Prior to joining Cal Poly Pomona's faculty, he was a postdoctoral researcher at the Washington University School of Medicine, and he earned his PhD from the Albany Medical College, where his thesis looked at how IL-12 alleviates the allergic lung inflammation in the absence of T-beta regulation. So Dr. Durant has been a pretty prolific, effective publisher. He is active in research here at Cal Poly, and we are now going to turn it over to Dr. Durant so that he can tell us about life and science in the time of coronavirus. As a reminder, any questions, save them for the end or throw them in the chat. So take it over, Dr. Durant.
Thank you, Katie. I usually for Science on Tap, we do we usually do it to, we hold it in a, in a bar. So usually we do a cheers. So I just wanted to cheers everybody. Everybody brought their a beverage of choice. If you guys want to do cheers, and with that, then with that, then I will go on. So today what I was kind of hoping to talk about is really about science and during this time of the pandemic and how science has, what the role is of science during a pandemic. And specifically talking about the COVID-19 pandemic, I wanted to highlight really some of the key findings that we have found in the short amount of time that we've been studying this virus. So it has been about 10 months since SARS-CoV-2 actually came onto we actually recognized that SARS-CoV-2 was infecting or was coming into the human population. And really in those 10 months, we've discovered an incredible amount about this virus and about this pandemic and the disease it causes. So I included these two quotes that were actually from different sources, and both of them really just highlight how the scientists across the world have come together to really address this issue that we're facing right now with COVID-19. And we've been set up a lot of clinical trials for vaccines and therapeutics to help to alleviate some of the impacts that this virus has had on our society. So kind of to highlight really what we've found, I'm going to answer these three questions. I'm going to address what is SARS-CoV-2, what is COVID-19, and how does SARS-CoV-2 cause COVID-19? And really the majority of my time, I'll probably be talking more about COVID-19 because that's what we are interested in, is how we can alleviate a lot of we're interested in researching how we can alleviate a lot of the impacts that the virus is having, the disease is having in our life. So first, what is SARS-CoV-2? So SARS-CoV-2 is a coronavirus. We've known about coronavirus since the 1960s. So coronaviruses in and of themselves are not something new to the virological field. We've known about them. They're called coronaviruses, and I'm sure many of you guys already know why they received this name, due to their shape under an electron microscope. And you can see here that you have these spikes that are on viruses, and it looks like it's a crown. And so you've already heard probably a lot about these spikes as well, these proteins that protrude off the surface of the virus, those are spike proteins. Coronaviruses do impact the respiratory tract as well as the GI tract, and they do cause respiratory infections. And generally there are the common cold coronaviruses that generally can be benign, or they can be just a common infection in the lung. But obviously we know that there are some coronaviruses that can cause severe disease. For example, the first SARS virus that we've emerged within the human population. So SARS-CoV-2 got its name from the disease it causes. So it does cause a severe acute respiratory syndrome. And since this is the second one that has emerged into the human population, SARS being the first one, this one received the name SARS Coronavirus #2. It is an enveloped virus, and it is a relatively larger virus compared to other viruses. And it is the agent that actually causes the disease, and the disease is COVID-19. There have been other coronavirus outbreaks. Again, you have probably heard of these multiple times as they've been as this virus has been in the news. We've related it to other viruses. So SARS is the virus that emerged in 2003. It was around for approximately a year, and then it went away. SARS was severe, and generally once you infected with it, you showed symptoms almost immediately. MERS-CoV is a virus that is still around, and we've had these occasional outbreaks with MERS-CoV. This is the Middle Eastern coronavirus, and generally it's in the Middle Eastern countries that there's been outbreaks. And then beginning in 2019, in January 2019, there was the SARS-CoV-2. Some of the reasons why these viruses are emerging, or we see these pandemics or these outbreaks of these viruses, is due to primarily these factors. The biggest factor is population density. As we move into these areas of the world where there are not as many human beings, we're going to come in contact with more and more wild animals, and we're going to become exposed to the viruses that are endemic within those host animals. The other reason why we see outbreaks is because we do migrate across the world. And so we can bring a virus from one region into a new region of the world where you have these populations that have not been exposed to those viruses before. And then there's this love for exotic meats. So going into finding some of these bush meats can also expose us to some of these viruses that we potentially have not come in contact with before. So there have been studies to kind of look at where SARS-CoV-2, it is believed that the origin of SARS-CoV-2 is from horseshoe bats. SARS is also, the original SARS virus was also believed to come from a bat origin as well. Currently, we do not know, so the idea is that the SARS-CoV-2 virus that was in the bat population was transmitted probably to an intermediate species, an intermediate mammalian species. And then from that intermediate mammalian species, it jumped into humans. Currently, we do not know what that animal species is, but there are continued to do studies to decipher what that species is. But once that virus jumped into humans, then we became the vehicle for the virus, and we're the ones that are spreading it among the human population. I want to kind of get into what COVID-19 is, or the disease that SARS-CoV-2 causes. COVID-19, the reason why it is called COVID-19 is because it is the coronavirus disease that was discovered in 2019. A lot of the news reports, and a lot of us actually call this virus COVID-19, but it's not the virus. When we refer to COVID-19, it's not the virus, it is actually the disease that the virus causes. I've kind of broken down the disease into 3 different stages. The first stage is the pre-exposure phase, or the period, and this is really going to be the time before we come in contact with the virus. And I'll talk about some of the work and the research that's been done to address what needs to be done during this time to limit the exposure to the virus. Then there's the viral replication phase, and this is where the virus is actually actively undergoing a replication cycle. And so we'll talk about really what some of the research is about the viral replication cycle and some of the therapeutics that have come out from that as well. Generally, there's a symptomatic phase within this. And then once you get past the viral replication cycle, there's the inflammatory stage. And this is where, due to the viral infection, it causes this inflammation in the body. And it's really your immune system that is causing the disease at this stage more than what the virus is able to cause. So these are going to be the 3 phases I talk about, and I'm going to talk about the research that goes behind each of these phases. Before I get into that, I want to talk a little bit about how SARS-CoV-2 is actually transmitted. So SARS-CoV-2 is transmitted person to person. Again, you guys have heard most of this. You know that you need to keep a distance from people, etc. SARS-CoV-2 is transmitted via droplet transmission. So droplet transmission versus airborne transmission is really dependent on the size of the particles that are in the air. So SARS-CoV-2, the particles that it spreads in are in droplets. Those can be found, so people who are infected disperse these droplets when they talk or speak, laugh or cough. And then if you're in the close vicinity, that's how you actually in these droplets that others are expelling. There have been studies to kind of demonstrate how far droplets can be spread. And they measure this in meters. And so the reason why we say 6 feet is because most droplets will fall within 2 meters of an infected individual, or an individual that's spraying out droplets. And so that is really the reasoning behind the 6 feet perimeter that you're supposed to maintain from others. The other thing with COVID-19 is that there are asymptomatic or pre-symptomatic individuals who can also be spreaders of this virus. All right, so now, like I said before, the majority of the talk of my presentation, I really wanted to talk about how SARS-CoV-2 is actually causing this disease, the COVID-19 disease. And so first, I'm going to focus on this pre-exposure phase. And really the goal of science here is really to figure out how we can prevent the transmission of the virus to individuals who are susceptible. And really those individuals who are susceptible are all of us. We are all susceptible to this virus. It is no respecter of persons. And so really that is the goal of the research here, is how can we prevent this from occurring? So the first, really the target one of the first things is our face masks. Face masks are one way that we can reduce exposure. And there have been some studies looking at face masks and how effective they are. Probably one of the best, or one of the studies that are one of the more recent studies kind of looking specifically at COVID-19 and the disease was accomplished in Hong Kong. And so really the percentage of individuals who adhere to the wearing of face masks in public is relatively high. It was around 93 to 96%. And here what they did is they looked at all the different countries compared to the Hong Kong region. And they looked at the cases of COVID-19 or of SARS-CoV-2 positivity. And so all this begins on in each individual country when they first got first had their first positive case for SARS-CoV-2 and looked at the rate of or the accumulation of cases in each of these individual countries. And you can see that the Hong Kong region is actually the one that performed the best, suggesting that the wearing a face mask is having an impact on the spread of the virus compared to other nations where the adherence to the face mask wearing policies, if there were policies, were not as high as in this region of the world. And so really one of the conclusions from this is they said they stated that community-wide mask wearing would contribute to the control of this disease or the spread of the virus. So beyond face masks, there was physical distancing, which we also already talked about as well. Cleaning and general hygiene, hand washing your hands for at least 20 seconds. There's already been studies multiple times showing that 20 seconds is needed. The other thing is maintaining personal health. So since there are underlying conditions for SARS-CoV-2 and COVID-19, the idea is if you are maintaining those conditions or remaining healthy, then you'll be less susceptible to the disease, to some of the aspects of the disease. And also getting the flu shot will help clarify what type of disease you have if you do start to show symptoms of the disease. The other one I wanted to highlight to help us reduce the risk of exposure is testing. So there's been a lot of research into testing and a lot of modeling on how testing can help. This is a group that actually looked at providing testing for asymptomatic individuals, and they set up these modeling models to look and see whether that would help to prevent the spread of SARS-CoV-2. And here they're actually showing that if you were to achieve a 75% precipitation rate of individuals who are asymptomatic or pre-symptomatic, and if they were checked every 3 days, if you were to be testing every 3 days, then the spread of SARS-CoV-2 would be drastically limited compared to, this would be drastically reduced compared to areas where there are no intervention or no testing done. And so there are efforts right now to push out a rapid test, rapid antigen test for these for these tests that are cheaper than other exams or other tests, so that we can get this out into the public and reduce the spread of SARS-CoV-2.
So before we move on, we have a question in the chat that I think fits in really well here. And so this is from Frances Mercer, and she's asking, are there any earlier data on the effects of masks related to the SARS-1 transmission? And if so, is there any suggestion that that might be why East Asian countries were already more comfortable wearing these masks? And if so, do you think that was an early oversight in Western countries?
Yes, there are other studies. So I didn't look at, I didn't pull up those studies, but I know that there are studies with MERS-CoV. MERS-CoV, I think, was an issue in South Korea. And so there were some efforts that were put out definitely to adopt wearing of face masks. I'm sure that the data from that has been compiled, but I did not look that up. As far as SARS, I'm not sure about SARS, whether wearing of face masks was encouraged at that time. I can't remember, but I'm sure that there has been studies done after the event looking at areas where face masks were worn.
All right, and then just one more quick question before we move on too far. What type of testing were being done in these studies? If they were looking at RNA, was there a window of detection? And if so, what was that window?
Great question. So this study that I'm showing you, this is actually a modeling study where they were modeling the getting out these rapid antigen tests or rapid testing. And so I do not know for sure if they clarified which type of tests they were saying, but ideally they were saying that you test often and you test frequently and among asymptomatic people, then you'll be able to catch those who have the virus quicker. And so these are modeling studies. I did not include any studies where they looked at specifically RNA. So the RNA, which we're going to talk about, the RNA, those take a little bit longer. And so there is a turnaround time for that. This one's more antigen based, which would be quicker.
Okay, thank you so much.
Thanks. And then the other one, just the other idea for during this pre-exposure period is to prevent infection. And really the best way to prevent infection is vaccination, is the most effective way that we can actually prevent a viral infection from occurring within a population. And today, actually, there was news about the Pfizer study. They showed that their vaccine is 90% effective in their phase 3 trial. But I'm sure there'll be more information that is released about that study in the future. So we are getting close to a vaccine, and then which will help with this whole pre-exposure period. All right, so then the other question is like, once you become infected with the virus, what occurs after that? So I'm going to now highlight some of the studies that were done during the viral replication phase. The goal here really is to limit the virus from replicating and also to limit the virus from spreading within the host or within the individual that's been infected and also spreading to others. So part of this is kind of looking at the incubation period for the virus. And so early on, we knew the incubation period, that this was different than SARS. So SARS incubation period was a lot shorter, and you started to develop symptoms pretty rapidly after infection. But with SARS-CoV-2, and part of the reason with SARS-CoV-2 that we're having such a struggle in containing the virus is the incubation period can be anywhere from 0 to 14 days, and that there are individuals who are asymptomatic or pre-symptomatic that are able to spread the virus. When we talk about the viral replication, this is the time when you actually have to detect viral replication. And in general, this is the detection of the viral RNA. So these are the genomic copies of the virus. In order to, a positive test result for SARS-CoV-2 is a PCR test with a CT value that is less than 40. For those of you who are interested in the CT values, this is an RT-PCR test that is less than 40. Generally, when the test is shipped out, the place that is actually running the test will determine your CT value of that test. And like I said before, if it's less than 40, then you will be considered to be positive for SARS-CoV-2. But early on, there was a study kind of demonstrating, looking at this incubation window and what the average incubation time was for this virus. And so you can see that with SARS-CoV-2, the incubation time was anywhere from 0 to 14 days, as compared to SARS, it was a lot shorter, and then MERS-CoV as well is shorter. But really based on this study, they came out that the average time for SARS-CoV-2 is 4.9 days, so about 5 days that you will have, you will be infected with virus but not showing any symptoms, and then you will start showing symptoms. And then the symptoms, which you guys have heard most of these symptoms already, include fevers and chills, cough. Generally, these are flu-like symptoms. These ones are a little bit different. You do get this loss of taste and smell, and some people do experience nausea or diarrhea from that. So like I said, like the goal here is really to limit viral replication. And so one thing that already known about SARS-CoV-2 is that it is an RNA virus. And RNA viruses use a specific enzyme to replicate their genome. And remdesivir is a drug that actually targets that viral enzyme to stop it from, ideally to stop the virus from replicating its genome or replicating itself. And so there was a study done in monkeys to look and see what impact this drug would have in limiting the ability of the virus to undergo replication. And you can see that they look at the titer of the virus, that this drug, which is shown in the red, actually had an impact in limiting the virus, or had an impact in the ability of the virus to undergo replication. They looked at the viral load. Also, I think they looked in the BAL, so fluid in the lungs, and you could that there was a lower number of viruses in the fluid of the lungs compared to the animals that only received a vehicle or that did not receive the drug. And then they also looked at the lungs of these animals, and you could see this is a normal pink healthy lung compared to an animal that did not receive the drug. And you can see that there were these areas of the lung that were undergoing necrosis or damage within the lung. And when they looked under the microscope, you see that the lungs were actually filled with fluid and that there were a lot of cellular infiltrates. So all these purple dots within these histology slides are these cells that have come into the lung and are causing more inflammation in the lung. When you look at the animals that actually received the drug, that there was more airways that were open, there wasn't as much damage, and there wasn't as much swelling or edema present in the lungs. So this really definitively shows that remdesivir does have an impact in limiting viral replication. There are studies in clinical trials with remdesivir, and remdesivir has been shown to help, but it's most likely a timing issue. They really need to get it early when the virus is actually undergoing replication. The data does suggest that if you get beyond that viral replication phase, that it would have less of an impact on the virus itself. Other studies want to look at the limiting of the viral spread. And so the idea here is to have the use of monoclonal antibodies or convalescent plasma. Convalescent plasma is plasma that is derived from individuals that have been infected before or have been infected with SARS-CoV-2 and have recovered from the disease. And generally, they're going to have antibodies that are able to target the virus. Monoclonal antibodies are antibodies that you can actually design or you can develop that can actually target specific areas of the virus. So the virus actually, SARS-CoV-2, the spike protein actually binds to the viral receptor on the host cells or in the cell, the individual that's going to be infected. And then once it binds to that, the cell will actually take up the virus and bring it into the cell. And then once the virus is in the cell, it can actually start replicating. The receptor for SARS-CoV-2 is the ACE2 receptor. So the idea is that if you get these antibodies in there, you can actually block this virus from binding to the receptor, and that will actually limit the virus from actually being able to get inside the cell and undergo a replication cycle. So a lot of people have actually looked at this. They've actually mapped out the spike protein and actually done 3D modeling of the spike protein. And then they've looked at the antibodies from convalescent plasma, and also they're trying to design antibodies, and they're looking at where those antibodies will interact with the spike protein to limit its ability to actually interact with its receptor. And so here they've actually mapped out some of those sites that the antibody can bind, or where the antibody has the most effective will have its most effective impact by binding to these regions of the spike protein. So they're highlighting this area of the spike protein, and then what's shown in magenta here is those antibody regions that they hope to have an impact. And so these are the antibodies actually that the president received, the Regeneron. These are Regeneron is developing these monoclonal antibodies, as is Eli Lilly. I think they're also developing something along these lines. But I think those are in early stages of clinical trials. All right, and then finally, after the viral replication stage is this inflammatory stage. Really the goal here is to reverse or counter the inflammation that the immune system is actually causing to occur in the individual or the cells that are infected with the virus. So early in the inflammatory stage, there's the cytokine storm phase. You most likely have heard of this. Cytokine storm. Cytokines are immune proteins that get produced by cells to really, the idea of these cytokines is to induce inflammation and to activate immune cells and immune mediators to fight off an intruding pathogen. What happens in the lung is that when you get these cytokines, these inflammatory proteins produced in the lung, they can induce what is referred to as this acute respiratory distress syndrome. And really this is inflammation in lung that will cause respiratory failures or the inability of the lung to function normally. What happens is, so this is a list of a bunch of these inflammatory mediators that get produced, and also these inflammatory cells that can get recruited into the lung. And when they're there, they actually can cause damage to these cells that line the alveoli sacs or the airways in the lung. And then that will lead to edema or swelling, so fluid can actually come into the lungs, and it will cause destruction or the inability of these cells to work properly for gaseous exchange, etc., in the lung. And so this injury will break down those barriers, resulting in more fluid and less function in the lung. And so researchers have looked at this to look and see what are some of those cytokines, if they are overly expressed in the lung, and also specifically which inflammatory proteins are actually produced. And so they looked at COVID-19 patients within the ICU. And they also looked at COVID patients not in the ICU and compared them to healthy controls. And they did see that in these cells, so these are lymphocytes that they're looking in specifically, they did see a higher production of GM-CSF. So this is one of those inflammatory proteins, as well as IL-6, both in the ICU patients as well as the non-ICU patients. And so really from this, actually, there are some clinical trials that are actually underway right now targeting the IL-6 protein, see if it can reduce the expression of that protein. There are clinical trials for IL-1, and then also they're utilizing corticosteroids. And corticosteroids are a broad-based anti-inflammatory drug. They don't target any specific protein, but they try to dampen all inflammation across the board. And then beyond that, there's the secondary infection phase. So these are where pneumonia can be, because of the fluid that's coming into the lungs, it will induce a pneumonia, and it will become a breeding ground for bacteria or fungal cells. And so that's another, generally antibiotics are given to these individuals. And there is research that shows that SREs is one of those bugs, bacteria that will cause bacteremia. Another thing that's people in this high inflammatory stage is this hyperinflammatory phase that is occurring. And there's a lot of coagulopathies that are occurring. So there's blood clots that are forming. There's also this multi-system inflammatory syndrome in adults and in children that is occurring. And the thought was what is causing, or what is the pathophysiology behind this? One idea is that there's these production of these autoantibodies. So since the immune system is wrapped up for a while, it's leading to the production of these antibodies that target self-proteins. Some of those proteins are targeting nuclear material. So these ANA antibodies are anti-nuclear antibodies. So these antibodies are actually attacking the nucleus of the cells. There are anti-interferon antibodies. So they're actually targeting interferon, which is another inflammatory protein that fights off viruses specifically. And also anti-phospholipid antibodies, antibodies that are actually against the phospholipids on the cell surface. And so this is from one study where they actually looked to see, where they actually demonstrated that in COVID-19 patients that were life-threatening COVID-19 patients, that there was a higher production of these autoantibodies, so antibodies that are produced against self-protein. And so that's probably what's leading to a lot of these coagulopathies that they're seeing, these blood clots. And it's also probably leading to some of the symptoms that they're seeing, like arthritis and loss of blood pressure, et cetera. Asymptomatic individuals that were infected did not demonstrate this, and healthy controls, obviously, the levels of these autoantibodies were extremely low. And so once we know this, we can actually, hopefully we can target these things so we don't get these autoantibody production. And then recently, or back in July, they actually showed that some of the immune cells are actually becoming exhausted as well during this hyperinflammatory phase. So this is more in the inflammatory phase. And so here they're looking at activation markers on specific immune cells. And they're showing that in the patients that have COVID-19, you had higher expression of these activation markers. So you had cells, immune cells that were, you had more cells that were activated. And they also looked at exhaustion markers, and they also saw that the exhaustion markers in these cells were higher as well, meaning that these cells were activated, but they were becoming exhausted, and they weren't able to carry out their normal function. So they actually saw this for both CD8 and CD4 T cells. So this also might be leading to some of the fatigue that they're seeing, and also some of the other coagulopathies is that these cells are becoming exhausted due to this prolonged inflammation that's occurring. and so then I just want to highlight here at the end that really the cases of SARS-CoV-2 are increasing. The best way to actually bring this down is to focus again on that pre-exposure period. That's the best way to prevent disease, is really that early stages. And so the research is being done to actually help target or look into that. And then I just wanted to kind of state here at the end, generally I share this slide with my students in immunology, that one of the reasons why we study and do science is because there is quite a wonder in science. The human body is one of the greatest wonders of the universe. It is complex, it is delicate, and is elegant. And really, the immune system, it really demonstrates that. Because when we look at COVID-19, it really is the immune system that's causing a lot of the disease process. The immune system that's meant to help clear the virus. And so the more that we actually learn about that immune system, the more that we can actually help to target it and help to alleviate the disease that is caused by this virus. And so I just want to end with really with that. One of my favorite, a quote that I really love is that from the Bible, actually, it says that I am fearfully and wonderfully made. And I think no better time do we see that science actually has a role in this, in helping to define how fearful and wonderful the human body is in the face of a threat like this. And so I am willing to take some questions from that now.
So thank you so much, Dr. Durant. We actually had a couple of questions come in through the chat while you were speaking. So the first one, Eliza Mitchell, would you like to ask Dr. Durant your question? I'll go ahead and ask for her. Her question came in very early and she asked, is there research to support the claim that the A blood group has a different outcome in terms of the severity of the disease?
So I know that there was a question early on. I cannot speak directly to that, whether there has been further research into that. My notion is that the blood group wouldn't have too much of an impact.
Okay.
So.
And then Dr. Mercer has our next question. So Dr. Mercer, do you want to go ahead and ask Dr. Durant? You'll need to unmute yourself.
Okay, sure. I was going to ask about the ACE2 dependence on infection. So like, does the spike need to undergo a conformational change in order for the virus to productively infect the cell? Or is any phagocytic or endocytic event enough to get a productive infection?
So I don't think I had, so it doesn't show it, but there's actually two. So not only does SARS-CoV-2 have to bind to the ACE2 receptor, but there is a protease that actually has to cleave the spike protein for it to actually be taken up, or for it to bind to the receptor. There is research definitely into that.
So we're not concerned that opsonization can promote infection in cells that don't express ACE2?
I do not think that there has been any evidence of opsonization, but you, because yeah, I don't.
Other coronas.
Yeah, I don't think, as far as I know, I don't think there is an issue with other coronaviruses, but I don't know for sure. Because I don't think there's any, like, we might develop immunity to one of the common colds, but I don't think that makes us more susceptible to another common cold coronavirus. So yeah, I don't, yeah.
All right, Dr. Buckley, would you like to ask your question?
Sure, thank you. Thank you for the talk, Doug. That was really nice. I've been really intrigued by the testing that they've done, but I was wondering what your take was on the limit in terms of time and the sensitivity of the viral RNA using RT-qPCR, how that compares with the limit, again, time-wise and sensitivity when using the antiviral antibodies. So basically we have two flavors of testing going on right now. One for the antigens based on our antibody that we're producing, and then the viral RNA. So how long and how much RNA has to be floating around us for us to be able to detect that within a specific time window?
Yeah, great question. So the one test, the big test is the RNA test. That's the PCR, the RT-PCR test. That one definitely takes longer. But I think there is an RNA antigen test as well, where you can actually look and you don't have to amplify it, or I think it's a quicker amplification. Anyway, but the limit of detection for virus, I do not know for sure. Actually, I should, but I don't know the limit of detection. And I don't know if anybody knows that for sure. That's why it's set at a lower than 40 threshold.
Yeah, I guess my question is, if you get infected like today, how long after you get infected, assuming that the sensitivity is okay, how long after you get infected, they would be able to detect that RNA, right? Because
I think yeah, sorry, I didn't mean to. I think they're able to detect it within 2 to 4 days after you get infected. But again, I don't know if they know specifically when an individual got infected. So I don't know if that has been clarified.
Yeah.
So.
Yeah, that's true. There's a lot unknown. But anyway, thank you.
Our next question is from Dr. Steele. Dr. Steele, would you like to unmute yourself?
Sure. Great talk, Doug. I really appreciate it. I'm curious if you know of any genetic changes in SARS-CoV-2 that might explain why it's spreading so much better than SARS-CoV-1 or MERS.
So I can't speak to that directly, but I think you could go back and like, is it so spread with SARS and SARS-CoV-2 were different because SARS the symptoms were immediately shown, not immediately but pretty rapidly, and they were able to isolate those individuals. While with SARS-CoV-2 they're not getting immediately isolated, and there is that asymptomatic spread. So there's more people that are spreading the virus out in public.
Could that be due to a change in the antigenicity of the virus, like a change in the spike protein?
Very good question. I can't speak directly to. I don't know. But I would not imagine. So I would imagine. Well, actually, I don't know. I was going to say that if it's not that, it's going to be something about the cell, its replication cycle and what it's activating inside the cells. So what's detecting that there's a viral replication in the infected cells? How are they able to counteract the virus, if that makes sense?
Yeah, it should be straightforward to look at the genome sequences. I just don't know how different they are.
Yeah, I don't know actually how different they are from one another.
So we do have a question from Mike Mendez. Mike Mendez, do you want to unmute yourself?
So I actually read his question. Maybe I'll read it.
Okay.
The question is, when considering the spread and scope of SARS-CoV-2, any evidence that it was constructed in a laboratory, or is it more likely to be natural pandemic? And is there a way for that to be confirmed? So I actually used to have that information on here, but it is widely agreed upon that it was not designed in a lab. It had to be nature that developed this virus. There's no, I think the simple fact that not simple fact, but yeah, they've gone in and not gone in, but there's this evolutionary tree that they've done with the SARS-CoV-2 compared to the earlier viruses. So there's been a lot of phylogeny studies done with this virus to look and see really where its origin came from. And in those studies, you can effectively rule out that this was designed in a lab.
All right, we have another question. Do we know why bats are responsible for so many spillovers?
Thanks for the question, Andrea. So bats are a unique mammal. There are a lot of different types of bats out there, different sizes. They are mammalian like we are, so but they are also flying mammals. And so just the bat is a unique mammal. And why are they responsible for so many spillovers? I think in general that because for some reason they are infected with many viruses, but they do not they do not develop disease from a lot of viruses. And since we are invading into territory where a lot of these bats are, I think that's when we are just coming in contact with another mammalian host. And so that puts us in a unique position because they are mammalian. There are a lot of viruses, we are too. And so there's just area for spillover to occur.
Glenn Kageyama, would you like to ask your question?
There's a lot of evidence that there are many species of mammals that can catch COVID-19 from us. And this poses a real danger because if it gets into our pet population, then that could be a problem. But zoo animals have had it. They could transmit it to monkeys, other primates. But right now, so far, the only other species that can transmit it amongst themselves once they get it are I think, minks and probably ferrets. And so their mink population has had to be exterminated because of that. But hopefully it doesn't mutate to a form that can spread amongst our dogs and cats, because that would be disastrous. The other thing that one paper I read about bats is that they don't seem to develop the same type of cytokine, the inflammatory response that we develop, and so they can better survive the exposure to the COVID, and it lives with them, and that's why they can survive it and spread it. But that's just one paper that I read.
Yeah, like going back to bats. Bats are, like I said, they're unique. Their immune response to a lot of these viruses are very different than our immune response, and so that's why. With other mammalian hosts, this is always going to be I guess not always, but it is a concern that is present. Potentially that the virus jumping into a new host species, it will have opportunity to develop and mutate, and then it potentially could come back into us. Like I said, I don't know how. That's not the primary concern, I think, right now. But yeah, that definitely is a concern that it can jump from one host or one animal to another animal.
Go ahead, Mary. Go ahead, ask your question.
Thank you for the amazing talk, Dr.
Thank you, Mary. Good to see you.
I was just wondering if you have any insight into why some people are more susceptible to severe COVID-19 symptoms, such as people from the European regions. And would it be because of the gene mutations that leads to less effective antibody activities or less effective receptor of the immune cells? Because I have high cholesterol level, and I know like how the receptor can take up the cholesterol in my blood. So and also would that be a fundamental concern when getting the vaccine? Thank you.
Yeah. So that's always a question, is why some individuals are more susceptible to in diseases compared to others. I think generally it comes down to really when you came into contact with the virus, how much virus you came in contact, and what was your health status when you came in contact with that virus. Also, genetics definitely has a role, but to kind of tease out genetics is a tricky thing, just because there's always environmental factors that have an impact on genetic factors, and genetic factors that have an impact on environmental factors. So to tease that out is a tricky thing, but I do think one day we will be able to kind of tease those things out. There have been some studies that looked at genetic factors. They look specifically at some of the immune genes, immune-related genes, to see individuals that have any defects in those immune-related genes, or disorders, if you want to call them disorders, that has looked at. And I think there has been some studies with COVID-19 on that. Would that be a concern for developing a vaccine? Absolutely, and that's why you need, when they're undergoing the trials, they want to have a nice diverse population that they try these in. And with any medication, there's going to be individuals who either react adversely to it or where it's ineffective. And so, but that's why in development you want to get a diverse group of people to try the vaccine in. I hope that answered your question.
No, yeah, that answered my question. Thank you so much.
Okay, we have one last, we have time for one more question. Is there anyone who wants to ask Dr. Durant the final question of the evening? Okay, so I'll go ahead and ask this one on behalf of Nadine Carlton. The Pfizer BioNTech vaccine efficacy looks great. Do you think that the other vaccines, such as those from Moderna and AstraZeneca, will be as effective?
Great question, Nadine. And actually, I'm going to, we're hoping to have a vaccine expert or a vaccinologist come and talk in one of these Science on Taps about vaccines. I don't know a lot about vaccines. I don't know specifically what the Moderna versus the AstraZeneca versus Pfizer, the difference between them. So I can't really speak to which one would be more effective. So I'm going to actually defer that question until the spring, if it all works out.
Yeah, we will consider that to be the teaser for hopefully a spring 2021 Science on Tap. So please join me thanking Dr. Durant for a wonderful presentation this evening. Thank you all for attending. I hope that you have a wonderful night. And again, thank you so much for coming.
Thank you.